Okay, let's go ahead start. Today we're going to talk about our module 2A, the microscopic examination of the brain, part A. This is the learning objectives. So we're going to particularly take a look at our fluorescent microscope, Nikon E200. That's an expensive microscope. Okay, let's talk about microscopes. The microscopes have two functions. First one is to increase the magnification of the image. Second one is increase the resolution of image. With these two things, we'll be able to tell details of an image. However, the light microscope has detecting limit, and that limit is 250 nanometer, which means if two small particles, they are around 250 nanometers under the light microscope, we cannot tell which one's which. We cannot separate them. And this is because the light microscope has a limitation on resolution, not because it has a limitation on magnification. If you don't understand why, read the lab manual and then you'll be clear. Okay, so let's talk about the light path of a light microscope. For all of the light microscopes, it's basically a group of magnifying glass put together, and then the light of this settings coming like this way. So the light comes from the bottom of the microscope and then goes through a set of lens, and then the light will be focused onto your sample and it will pass your sample, go through your objective lens. So these are the objective lens and go to some side of mirrors and then reflect it to your eyepiece. And this is another set of magnifying glass. So the microscope that we're going to have in our lab is a fluorescent microscope. It is pretty fancy, but it's built up on top of light microscope. So pretty much all the fluorescent microscope are built up on top of a light microscope. So if this is a light microscope, you have light coming from this way, hit your sample, goes objective lens, get reflected to your eyepieces, right? If that is a light microscope. You can have something add on here. That is another light source, and this light source is usually a laser, which means have very high intensity light and go through this way, and there's a mirror. This light get reflected. Once the light hit your sample, is going to be reflected and then go back to your eyepieces. So basically for fluorescent microscope, the light source and your eyepieces are on the same side of the sample. So you'll hear people say fluorescent is epifluorescent. Epi means above. So that means the light is above your samples. For light microscope, your light is underneath your sample. So let's talk about fluorophore, because without fluorophore, fluorescent microscope will not work. So fluorophore is a special chemical molecule. They can absorb certain wavelengths of light and emit another wavelength of light. So the light this fluorophore absorb, we call them excitation light. And this light will have higher energy than emission light. Here's how do I remember that. So the fluorophore will eat higher energy light and poop out lower energy light. Okay, so how do you know a light have higher or lower energy? So a light is a wave of photon. If you don't understand what is that, you can ask your physics teachers. Basically, it's saying light is a small ball, it's bouncing around, and because the wavelengths, so from this peak to this peak, we call that wavelengths. Because the wavelengths of different light is different, so you see different colors. You see the purplish light here, that light has a shorter wavelengths, and the red light have bigger wavelengths. Okay, so I labeled wavelengths for different color of light. 400 nanometer is a purple light. The purple color light, which have small wavelengths, 400 nanometer, will have higher energy. Longer wavelengths light, which is red, have lower energy. It is a little bit counterintuitive, right? You would think red will have more energy, purple or cold color light have lower energy, but it's not. Okay, so how does fluorescent microscope work? The most important thing is this tube here. There is 3 compartments. The first one is a filter. We call them excitation filter. This filter will filter out all other light except the excitation light. And then there is a mirror. We call them dichromatic mirror. This is also called a half mirror. What is fancy about this mirror is this mirror will reflect light with higher energy, but will allow light with lower energy to go through. So in this situation, this fluorophore will absorb green light and then emit a red light. Red light have lower energy. So for this half mirror, the higher energy light, which is green light, will get bent, reflected, and then the red light will be able to pass through. And before you see this red light, people put another filter here. They usually call emission filter. This filter will further filter out other light, will only let emission light going through. So this way your image will become cleaner. Okay, so let's talk about the classic fluorophores. So most fluorescent microscope will set up the light and then the filters based on these three classic fluorophores. So first one is DAPI. That's used in histochemistry a lot. We're going to talk about how DAPI works just in a second. And then FITC and TRITC. So this is the excitation and emission light for FITC and TRITC. So they have to be used together with antibodies, and they can be used in immunohistochemistry. So if you incubate your tissue with FITC or TRITC, they are not going to stain your tissue. So DAPI works differently from FITC and TRITC. So here is the DAPI. I mean, there's a whole chemical name for that. I don't know even how to pronounce them. Okay, so how does DAPI work? They will bind strongly to AT-rich region in DNA. So this is a DNA, and if you find that DAPI is binding to there, that means there are a lot of ANT nucleotide in there. So DAPI only bind to AT&T, not to verizin. What is special about DAPI is it can stain both live and fixed cells. So if you put your finger in the DAPI solution, if you put yourself in a violet light, your finger will turn blue. Is that a good thing? No. Why is that? Because if you have something stick into your DNA, that will cause some mutation. Mutation means your cells are going to go crazy. You will have some risk to have some cancer. So you don't want DAPI to get onto your skin. Particularly, you don't want to eat DAPI, and you don't want DAPI to get into your eyes. However, it's not very effective in staining living cells. So which means if you stain living cells with DAPI, you probably have weaker signals, but they do work on living cells. So here is an example of using DAPI with other fluorophore together. So the blue one, that's a nucleotide, that's DAPI. And green part is FITC. Conjugate FITC with antibodies. So this antibody will bind to microtubule, so that's a FITC. And then actin was stained by TRITC. That's a red one. TRITC was bind to antibodies, so they will be able to stain actin. Again, if you just stain the cell with FITC and TRITC by itself, you are not going to see anything. They have to conjugate, which means attached to antibody to stain. So here's another example of how fancy the fluorescent can give you. So what do we have here? This is a vessel in the brain. So the cyan color is the vessel wall. And then you see this very green thing here, that is astrocyte. And what is fancy about this astrocyte is, if you gave some signal, they will do something. Okay, did you see that? If you signal, the astrocyte is going to squeeze the vessel tight. So that's how does your astrocyte regulate your blood flow in your brain. Okay, let's talk about the benefit of using fluorescent microscope. Other than using fluorescent microscope, you can have a choice of using pigment to stain your sample and take a look at your sample under a light microscope. So using a fluorescent microscope, you will have the first two benefits. First one, you are going to have increased sensitivity because your noise is going to be low. And that is because all the fluorescent stainings, your background is black, it's dark. So there's almost no noise at all. That's how you increase your sensitivity. And for fluorescent microscope staining, the steps are usually very fast. That will save you time. And both the pigment staining of fluorophore stainings, you will have these three benefits. First one, you can multiplexing, which means you can stain different structures with different dye, with different colors at one time. So we call this type of staining multiplexing. For fluorophore, you have more choice in terms of colors than using pigment. And fluorophore works on living cell better than pigment. Some pigment also work on living cell, but fluorophores usually work better. And both of these techniques, you can quantify the molecules that you are trying to stain. Usually, the more colorful region, more brighter region means you have more amount of the protein or the molecule that you want to quantify. Okay, now I have some questions for you. First one is, will you see dendrite and axon after DAPI stain? The answer is no. Why is that? Because DAPI only stain AT&T rich region in the nuclei. So the dendrite and axon don't have any nuclei. Am I right? Okay, the second is, can you tell the shape of a cell after DAPI stain? The answer is no. Why? Because you can only tell the shape of a nuclei. You cannot tell the shape of the cell after DAPI stain, because DAPI only stain the nuclei, right? Okay, so you can't tell the shape of the cell. Why do you use DAPI? So here's a question. Can you tell the structure of a different part of the brain after DAPI stain? And the answer is yes. The question is how. So here's a clue. If you can only label the tall buildings on the map, and can you tell where is Roanoke, where is Krisenberg, where is Blacksburg? The answer is yes. How? By telling the density of the tall buildings, right? Roanoke will have a higher density of tall buildings. And come back to our question. So different part of the brain will have different density of nuclei. So that's how you tell the structures after the DAPI stain. Okay, now let's talk about our expensive fluorescent microscope. This guy worth $20,000, which means if you steal that, you can buy a new car, right? Okay, let's talk about the light path of this little guy. First one is a fluorescent light path. So the fluorescent light is generated in this black box. Then the light will travel along here, and there is a dichromatic mirror will get bent. So this light you generated is excitation light. And then the excitation light will go through our objective lens and will hit our sample. So sample is here. And then once the sample absorb our excitation light, will emit emission light. The emission light will go back to the objective lens and travel back here, and there is dichromatic mirror, the half mirror. Because this light is lower energy, will go through the mirror and go to your eyepiece. Or if you want to see your image under the camera, you can send a signal to the camera. And the other light path this microscope has is the bright field light. So the bright field light is for the light microscope. So underneath here, there is a light bulb and there's a bunch of lanes. These lanes are used to focus our light, so the light can be focused to our samples. So the light path for light microscope for the bright field light is coming this way and hit your sample and go through to your objective lens and travel all the way to your eyepiece or to the camera. Because there are two light paths, there should be two power buttons, right? Yes, there is two power buttons, and both of them with a dim. So here is the fluorescent power button. You can see there's off, and then you have choice of different colors. You can have white light, red light, green light, cyan, violet light. And you can change the intensity of the light. That's a fluorescent power switch. And this one is for bright field switch. If you turn it off, the light will not coming from this direction. You can also change the intensity of the light by turning this knob. Okay, there's something special about fluorescent microscope. There are curtains. It's only me call them curtains. I think that's the best name for them. Nobody else call them curtains. I don't know why. Okay, let me show you what are the curtains. By the side of a microscope, you see this bar. If you pull the bar out, you are going to close the curtain, which means there is going to be some light block that will block the light coming from this light box to your sample. And if you push that bar in, the light path will be cleared out. So you're gonna have light coming through. Same thing for bright field. You have this cap. If you put cap on, the light will not be able to go through. And at the same time, also if you put cap on, it will also prevent the light from the environment hit the lens here to reflect to your sample. So this is the lens that help us to focus the light to our sample here. And underneath this lens, there is a light bulb. So the curtain for bright fields is here. So the question is, why there is curtains? If there is a power switch, just turn off the light, right? Usually we use a curtain in the middle of the experiment. We use that to block light. Because turn on and off the light too often will reduce the lifespan of the light bulb. So that's why we want to use curtain to temporarily block the light instead of turn off the power switch. Obviously, the microscope light bulb is expensive. For this microscope, there is something special, which is there is a camera mounted to the microscope. This is a camera, this white box here, and the camera is connected with a monitor, which means if you want to take photo of your image, you can just use this camera. However, you have to switch between your eyepiece and the camera. So you see there's a bar here, there's a label. Bino means eyepiece. If you push the bar in, the signal will send to your eyepiece. If you pull the bar out, the signal will send out to the camera and it will project it to the screen. However, you cannot see your image both on the eyepiece and the camera together. To turn on the camera, you have to go on top of the camera. So that's here. You have a power switch and there's an SD card that you can storage your image and you can transfer that to your computer. And here is the button for monitor to turn the monitor on. Okay, let's take a look at the microscope lens. So this microscope have 4 objective lens. So the lowest magnification is 4x. And then you have 10x, 20x, and 40x. Because our eyepiece is 10x, we have 40, 100, 200, and 400 magnification. So how do you calculate the magnification? Is the objective lens power times the eyepiece power. And there's a platform, which is this black guy with the clip. We call this thing stage. So we put samples on the stage, and we can control where the stage go by turning the knob. This is the stage. We put our slides on this clip. So when you're moving the clip, you'll be able to slide in your slides and clip here. And when you need to move the slides up and down, back and forth and left, right, there are knobs that control the stage. This big knob here is moving the stage up and down. So move the stage up and move that down. And there's this fine adjustment. You wouldn't see much. It's moving up and down very small amount. If you need to move the stage back and forward, there's a knob on this side. So the upper part will move the stage forward and backward. And then the knob underneath that will move the stage left and right. So that way you don't have to use your hand to move your slide. So when you're looking for samples under a microscope, you always want to start with 4x objective lens because this one will give you a larger area of view. You can find things a little bit easier. And you always want to start with the highest position of the stage. Why is that? Because that's less moving. Because the best focus plane is close to the highest stage position. So which means first of all, you only need to move one direction, which is lower the stage down, right? And then second, you don't have to move that much. So work smarter, not harder. If you don't listen to me, when you're getting into a microscope, you are going to have a lot of stress. If you follow these 2 directions, you're going to feel pretty good. Okay, we make a lot of brains, coronal sections from a mice brain. Before we stain them or look at the structures, we have to sort the anterior and posterior. Why is that? Because certain structures only exist on certain slides. If you keep searching on the anterior slides for a structure that's supposed to be on the posterior slides, even you work so hard, you still don't have any result, right? So that's why first thing we have to know which one is anterior, which one is posterior slides. Okay, so here's are all the slides that we make out of one small tiny mouse brain. So the first slice we cut it, coronal section is from roughly from here, and then the last one is rough from here, which means anterior to this part, all these things were not included. Anything posterior than this one will not include it, which means we don't have any slice that will include cerebellum. So all the slice that we have between this cut and this cut, those slice looks pretty close to each other if you first time looking at them, right? Okay, let me tell you how do I sort them out. Okay, I categorize them into 6. So there are 6 categories of them. So if you take a closer look of them, the first row look pretty much similar from each other because the slice is 50 micron thick. So the adjacent slice belong to similar structures. So even this slice and that slice, they are 500 micron away, which is half millimeter. So they still share the similar structures. Let's take a closer look of them. So the first one, the first row, I think they look like the Simpsons mother. So this is the hair. So there are hair, the puffy hair, and then this is the face. So that's how I remember the first row. They look like Simpson's mother. If you don't know who's Simpson, Bart Simpson, right? Okay, and then the second row look like Bart Simpson's father's face. Okay, because that's like oval shape, and there's nothing like puffy because he doesn't have hair. Okay, like to Bart Simpson's father. Look, the whole row, right? Okay, and then the third row, I think that looks like the Kung Fu Panda because see this triangle shape, that's his eye, and then there's a big cheek, so that's his cheek, and then there's 2 dots, that's his nose, and there's a little mouse there. Okay, so that's the third row. And then the fourth row, I think that looks like angry, grumpy Garfield. So see that little white thing here? That's actually the fornix. If you forget what is fornix, that belong to the limbic system. We identified them in module 1, the sheep brain, right? So that's the fornix. So that looks like the fornix, right? Okay. And even the first one, the fornix is a little bit shorter, but still look like the grumpy Garfield. Okay, so that's number 4. And category number 5, that look like Dora, the explorer, right? Why? Because see the last one, this one has kind of like darker color. Eye look like structure that look like her eye, right? And even the first one, there is still a very small eye look like structure. Actually, this structure is hippocampus. Okay, that's I categorize them into 5 because all of them have this eye look structure in there. That's a hippocampus. Okay, the last category is Dora with crazy eyes. Why? Because the eye get bigger and longer. Okay, so that is still hippocampus. So remember, hippocampus is a long structure that's wrapped around the brain. Okay, so all of these rows has long hippocampus in there. Okay, so from the anterior to the most posterior ones. So at the beginning, you don't have any hippocampus, and then until you reach here, you start to see a little bit of hippocampus, and you know these slides are posterior. Okay, so here is the summary of the 6 categories. At the beginning, you don't have much structures. You have this puffy hair look structure, look like Simpson's mother, and then Simpson's father. Kung Fu Panda, you have this big cheek. You have this hole here. Actually, this cheek is caudal putamen, and that's the lateral ventricle. And then you start to see fornix, and then hippocampus, and then hippocampus get longer. So from the anterior till the most posterior. Okay, so just in case you may wonder why some slices are broken. See, there's broken thing here. It's broken here. That's because when we're cutting the slice, we happen to tear them up. When you see these things, it doesn't matter because when we're taking look at certain structures, we're really looking at very small structure, like maybe here or maybe here. So the tear really doesn't matter. We can have the other side if you want to see the structure here because the brain is symmetric. Now let's talk about how to find certain structures on the brain slice. So you have to use the brain atlas. First thing before you start, you have to match your brain slice to brain atlas. So this is the brain atlas. There are a total 133 images. So that's the image, and this each each image has a number. The smaller the number means that slice is more anterior. So in this situation, this image 32, and that's obviously more anterior than the next image, which is image 33. Okay? Okay, let's match the image here. So the first one that we got in our lab is the Simpsons mother's head, right? I think that's a 32 match to this image a lot, because first one you have this half puffy thing here, and you have this lower part, and you have a little bit white thing in here, and that's a white thing. That's actually the anterior commissure olfactory limb, the ACO. Okay, and the next one, this one looks the image 43 a lot, because you have this white dramatic thing here, and you also have white thing in here, and you have a dot in here, you have a dot in here, and actually this white thing is corpus callosum. Okay, and see the Kung Fu Panda. So the big cheek here, so pretty uniform structure, and then you have this corpus callosum, you have this corpus callosum, and the hole there is actually the lateral ventricle. Okay, and then this one matched number 60 a lot, because you have this fornix here, and the fornix, the shape match. The white thing here doesn't match too much with here, but that's the best one I can find among all the images that Brain Atlas gave us. And next one, the Dora, matched number 70, because the hippocampus here, you see the hippocampus here. Okay, and then the darker region is because there is special area that have a lot of nuclei, so that's why you see a lot of stain in here. We're gonna see that after we do the DAPI stain. And then the Dora with crazy eye here, that is the hippocampus here. So that image is 79. I think the 79 matched this one the best, even though we have some tear around this slice. Okay, let's talk about how to locate certain structures. So first one, you open this Brain Atlas, and then there is a search bar. You type in the structures that you want to search. For example, corpus callosum, you can type in here, and then you have to click the one that Brain Atlas want to use. The way that you type, they doesn't like it. Okay, so you click that, and then it's going to highlight the structure in purple to tell you this is a corpus callosum. It's going to highlight the structure on every image that have this structure. Okay, let's take examples. First one, let's locate the hippocampus. Okay, so I type in the hippocampus, and then on image 32, I don't have any purple. 43, purple here. 49, purple here. 60, you get a long structure. That's the corpus callosum. 70, corpus callosum. Okay, 79, corpus callosum. So if we go back to our brain slice, you should know which one doesn't have corpus callosum, right? It's pretty easy to tell, because this one has white thing here, this one is white, and you have this white band structure. This one is corpus callosum. The only one that doesn't have this one, right? Okay, next one. To locate anterior commissure, the olfactory limb, for sure we call it ACO. So I typed that thing in, and then the ACO was highlighted. So here it's ACO. If you use the other side, that is the this white part, and same here. There is a little bit white in here, and same on this one. This one don't have any purple, right? It means this probably the most posterior slice that have ACO in there among all of our slides in this image. This one don't, this one either, right? Okay, let's take a look which slide we cannot find ACO. Okay, so this one's ACO, the white here, right? And this is the white ACO, this is the ACO. That's not ACO, because on this image we don't have ACO. This one does look like ACO a lot because it's a white dot, right? However, if you match our Brain Atlas, that's number 70 image, there is a little bit white dot here, but that's not ACO. Same here, the white dot here, that's not ACO either. There's some other structures around here. Okay, next one. Lateral ventricle. For the first slice, there's a very small tiny lateral ventricle in here. Matched to the other side, that's this guy here. You probably even don't see it. Okay, and from the 43, you will see a lot of big lateral ventricle, like a crack. And there's this one, the lateral ventricle, and then when we get to image 60, the lateral ventricle become bigger, and 40 and 70 become this one. It's the lateral ventricle, and 79 even you still have lateral ventricle in here. So on this side is somewhere in here. Okay, now on this image you'll be able to tell which one you can find the lateral ventricle. So you should be able to find a lateral ventricle somewhere in here, but it's very small. And then this one, see the darker place, that's the lateral ventricle. And Kung Fu Panda's eye, lateral ventricle. And you have lateral ventricle in here. And for this one, this is the lateral ventricle. And here, you see the hole here, that's the lateral ventricle here. Okay, next one, nucleus accumbens. So nucleus accumbens belong to deep brain nuclei, and that contribute to fine motor movement. If somebody have Parkinson's disease, their nucleus accumbens probably also be damaged. Okay, so on 32, we don't have nucleus accumbens. We only find nucleus accumbens on these two images, right? 43 and 49. And want you notice that nucleus accumbens is actually around the ACO on 43, and on 49 it's on the side of the ACO. So the nucleus accumbens really don't have distinct edge around it. So you have to use the brain atlas to roughly estimate where the edge is. Okay, so on these slides, you know where to find the nucleus accumbens. This one we don't, this one we don't, this one we don't, this one we don't. And will find a nucleus accumbens around the ACO, so somewhere in here. And if the image looks like a Kung Fu Panda's face, that's somewhere in here, the nucleus accumbens. Next one. Hippocampus. So on the brain atlas, they call the hippocampus hippocampal region. Okay, so if you type hippocampal region, they're gonna highlight the hippocampus for you. So on 32, you don't have any. On 43, there's a very tiny small part of hippocampus. 49, there is a small part. Image 60, same thing, small, very small. And 70, you have a big hippocampus. So the eye of that Dora, hippocampus. And then if you go more posterior, Dora's eye become crazy, and that's a whole hippocampus. Okay, so now it's easy to find hippocampus on here, right? So on number E, just there, Dora's eye, that's the hippocampus, and the Dora's crazy eye, hippocampus. All right, let's talk something crazier. Hippocampus can be further divided into substructures. So hippocampus has 3 CA1 regions and 1 dentate gyrus region. So if you highlight the CA1 field, the brain atlas is going to label CA1 purple for you. So that's on image 70. This part is CA1. And if you highlight CA2 and see there's a small part of CA2, and that is CA3. If you highlight the dentate gyrus, that's the dentate gyrus here. And that's something happens in image 79. So same thing, CA1 here, CA2 is here, and CA3 is here, and then the dentate gyrus. Okay, let's do more crazy things. You know what? CA1 can be further divided into layers. Let's take a look of this layer. The layer has a lot of nuclei here. Okay, so the darker region here, if you highlight CA1 pyramidal layer, it's going to highlight that layer for you. So this layer is the CA1 pyramidal layer. And same thing on image 79. So that's the CA1 pyramidal layer. And then the dentate gyrus also have different layers. All right, so if you highlight the dentate gyrus granule cell layer, this peak look structure is going to be highlighted. That is the granule layer of the dentate gyrus. All right, finally we're done. Okay, so next one, we're gonna go to the lab, and I'm going to do the demo of the brain slide staining, and we're gonna look for structures under the microscope, and you are going to see some beautiful images. All right.